So good morning. It's a pleasure to be part of this exciting program. What I'm going to talk about today is how an informational set of molecules, RNA and DNA, which we have studied for decades, have now become therapeutic agents that we can begin to probe diseases and treat diseases. Now, I am a member and a founder of Alnylam Pharmaceuticals, and I'm going to use some data from Alnylam Pharmaceuticals to make a point today. But I would say that the science that established the biotech industry was primarily focused on interferons and protein hormones in 1980, 1990, monoclonal antibodies in the next decade. Oligonucleotides became exciting around the turn of the century in 2000, 2010 with siRNA and microRNA, as I will explain, and then CRISPR and cell therapy. But the interesting one is at the bottom, and that is gene therapy. Gene therapy has been a possibility for the last 40 years, and it still is something being developed because it has this very large effect of possibly having off-target activities that are important. But we're going to talk about the oligonucleotide and siRNAs. Now, we've frequently heard that the sequencing of the human genome was a great media event, but where's the beef, as they say? Where's the impact? And the impact has come in part out of these oligo and RNA therapies that could not have been developed without this advance. And what we see now is a massive decrease in cost in sequencing the human genome. This has enormous implications and is actually the basis of our design of treatments for cancer and many other activities. And it has also brought together what I think is the most important development of the future is the bringing together of IT and molecular and cell biology. That wedding is beginning to come out of the advances of high DNA sequencing. But one specific example I want to talk about as just an example for a whole field is genetic diseases. This is from 2015, and you will see at the top there were about 3,000 Mendelian disease genes known. That's probably close to 5,000 to 6,000 now. 8% of all live births have a genetic disorder that is apparent in adulthood. Estimated each child with a genetic disorder costs about $5 million in healthcare, loss of income, other societal costs. And the diagnostic rate of genetic disorders in 2015 is about 11% as a child and 34% as adult. Those numbers have changed slightly, but not significantly over the last 10 years. But we will find the time in the future when everyone will essentially be sequenced. Well, let's talk about one example, which I think is a breakthrough in genomic information being applied to treat a specific disease that's a genetic disease, and this is Emma. Emma is a patient with spinal muscular atrophy, and she was treated with an antisense oligonucleotide to shift splicing, and she can now walk. And many children who have spinal muscular atrophy of a severe type do not live beyond the first year of age. They have defects in development that are almost irreversible because they lose neurons. So Emma benefits from a treatment that is designed on genomic sequences and delivered through a specific, interesting way. While spinal muscular atrophy is due to a defect in this gene, spinal motor neuron 1, is deleted or irreversibly mutated. Patients miss this protein. Motor neurons are defective. The patient loses control of their lungs and others and succumb. In the genome, there's another gene next to it, spinal muscular survival motor neuron 2 that differs from 1 by having a variation in one nucleotide sequence. And what happens in this gene is that 6 is spliced to 7, spliced to 8 to make a functional protein. And in this gene, 6 is spliced to 8, 7 is skipped. The protein is unstable and it's degraded mostly, and therefore you only get 10% of activity. This is a genomic discovery. And what makes it particularly genomic is it was analyzed by Adrian Krainer, which I'll acknowledge in a moment, to be due to that mutation, which I illustrated before, a variation in exon 7, but he discovered in the downstream intron that there was a protein that bound here that suppressed 7, and therefore by using an antisense oligo injected into the spinal column of a baby, moving through the spinal column through cells, entering the cell, going to the nucleus, binding to the RNA, you can shift the splicing by blocking this sequence to 6, 7, and 8. And that's now used in children less than a month old. And it promotes their development normally. So here was a breakthrough, a genomic breakthrough, oligonucleotide central nervous system, a benefit by perturbing splicing, the most integral part of the gene expression system in these neurons. And that was done by Adrian Krainer at Cold Spring Harbor. He discovered those sequences. He collaborated with Ionis, Frank Bennett there, and Biogen licensed it and took it to fruition. Now, at the turn of the century, there was other advances that changed oligonucleotide approaches to treating disease and amplified the impact of the genome on therapeutics. And that's from the discovery of Farr and Mello, where they discovered RNAi in 1998 and got a Nobel Prize in 2006. And what they specifically discovered is that double-strand RNA transcribed from a gene, much like we make Moderna message, but now we make double-strand. You can introduce it into a worm, and it will be cut up into little pieces called SI, small interfering RNA, and it will silence a gene from which that RNA is made. Sequence to gene to silencing. And that led to the idea that this could be a therapeutic modality where you could take a genome sequence and design a therapy if you knew the gene, when silenced, would be beneficial. Well, it turns out nature made this much easier to do in our cells, because shortly after this discovery of RNAi, again in 2000, it was discovered that our genome codes for 500 to 1,000 unknown genes at the time. These genes make small RNAs called microRNAs. They pair with almost all the RNA message in a cell in combinations and regulate that RNA in terms of expression. So here in each of our cells, every cell we know of in the body, we have a professional expression RNA regulatory method. So the siRNA is this intermediate. And when you take those synthetic double-strand RNAs and introduce them, they enter the cell in this pathway, pair with proteins that are carrying out this function in the cytoplasm and giving you a message. So this led to the possibility that we could harness this natural pathway to basically introduce into cells an oligonucleotide to treat a disease, silence the message, cleave the message, and be catalytic and do it again and again. So when this became apparent, Alnylam was established, and the question, which has been there for as long as nucleic acids were known, DNA and RNA, is delivery, delivery, delivery. It's wonderful to have something that'll silence a gene, but if you have to get it through the membrane of a cell, The question is delivery. And delivery has been approached in a number of ways, but I'm going to talk about delivery because it is the essential step between RNA, nucleic acid, and gene. And here in this case, it's lipid nanoparticles, where the RNA is encapsulated into a lipid nanoparticle. It's made with a lot of lipids and PEG and other components. In the system, it pairs with ApoE. This is the system ultimately an ilum developed. It enters the vicinity of an hepatocyte. It's internalized. The pH shifts. The lipids change in conformation, releasing the components of the nanoparticle into the cytoplasm. So this was known in 2000 as a possible pathway. The issue is how to make it therapeutically possible to use. Because if it's not efficient, you have to deliver a large amount of the LNP particle that itself activates complement, and you have to deliver a lot of nucleic acid to the system, both of which have non-specific, what we call off-target relative to these effects. So what happened over about 5 years is that the efficiency of this system was increased 3 orders of magnitude. And that made it possible then to treat disease. So this is in the liver. This is transthyretin. It is primarily in a genetic sense, a mutation in the TTR gene that makes a misfolded protein. That protein is made in the liver. It is secreted into the blood, taken up by motor neurons in the system, and those motor neurons ultimately are destroyed. This is an amyloid disease produced by one organ, the liver, captured in neurons and destroy neurons. All right? Just think about another amyloid disease we'll come to later, and that's Alzheimer's. So how to treat this disease? Well, you can take an LNP with an siRNA, introduce it into patients, and follow silencing. And this is a placebo, and this is a treatment. And you notice this goes out over several months. Patients have been treated with this 6 or more years. It is a very long-term treatment for hereditary HTTR. Now, this same technology, which is used now for therapeutic treatment of a liver disease, is the same type of technology, very similar technology, that's now used to deliver messenger RNA in our Moderna and Pfizer type vaccines. So the technology of lipid nanoparticle delivery to cells has been developed over this possibility of using RNA and nucleic acid as a therapeutic modality. But the lipid nanoparticle is not the only therapeutic modality. In fact, this is much more convenient. This is taking an siRNA and tagging it covalently to a GalNAc set of sugars. Turns out that on your liver cell, there's receptors for those sugars. They then get bound. The siRNA is taken in. It is ultimately released from these endosomes and enters the cytoplasm and silences the gene. Now, the thing that makes this convenient is it can be administered sub-Q, where liquid nanoparticles are primarily administered therapeutically by IV. And the other is this RNA is now available to be modified. And if you modify it and you deliver it and other activities, you can increase the efficiency another 3 orders of magnitude. And the exciting thing is shown here. This is PCSK9, protein produced in the liver that controls cholesterol. And these are human patients dosed with one increasing dose, 100 mg or 300 mg. And you see silencing one delivery sub-Q for 6 months. That's amazing. I can inject the patient, and for the next 6 months, that gene is silenced. We don't have to worry about compliance. We don't have to worry about them taking a pill every morning. They get silenced because of this activity. This has now been approved in 2 therapeutic modalities by the FDA. It's used to treat oxalate-type disease, a porphyria. It is a treatment of PCSK9 that Novartis has purchased and bringing to therapeutic development. A striking example of delivery to tissue, specific tissue in the body. But I think when we look forward, you combine these 2 stories. You take the oligonucleotide shifting SMN1 or 2 and siRNA, and you say, what could happen in the central nervous system if this technology works? So the technology does work. It's not in humans yet. You've got to think about all the things that's different between an idea and a reality. But if you think about some of the major unmet needs, Alzheimer's, Parkinson's, frontotemporal, Huntington's, ALS, and others, all of those have now been identified to specific genes. And if you got a specific gene, you got a specific oligonucleotide siRNA that could silence it, if it would be beneficial. The issue is delivery. Well, what has happened? This is delivery in rat, and it's delivered by injection here in the lumbar region, and then after a period of time, tissue is taken, and this is the SOD1 gene, and it's being silenced by an siRNA conjugate injected here, and you see about 90%, 80% silencing in cells from this region at the RNA level. You see about 80% in this thoracic region. You see about the same in the cervical region. You see about the same in the cerebellum, and you see significant changes in the frontal cortex. This is in a rat. So it's very exciting in the idea that one could use these delivery technologies to modulate issues in the CNS. Well, you can do it in non-human primates. This is an example of, in non-human primates, an siRNA to APP, which is the amyloid-forming protein in Alzheimer's. And this silencing is within cells in the Alzheimer, not a secreted protein, within the cell in which the amyloid forms. And what you see here is about 75% silencing in the cervical lumbar region. Cervical, it's a little less. All the way into prefrontal, you get significant silencing. So, and you can do it in the eye. So here is ocular TTR protein knockdown due to injection of siRNA in non-human primates, and you see about 90% silencing. So with specific delivery technology, you can take siRNA and silence other genes in the CNS. It is a delivery challenge. The biochemical machinery is there in every cell as far as we're aware. So I think this blending of the discoveries of RNA, DNA historically, the Human Genome Initiative giving us sequence to identify genes and genetic structure, the development of oligonucleotide antisense technology, the development of siRNA technology has opened up, with delivery to specific cells, opened up a whole new way to approach therapeutic treatment of patients. And I thank you for your attention.
Thank you so much, Professor Sharp. That was great. We can take some questions from the audience. Just let me know if you have, and I'll bring you a mic.
Hi. I wanted to know if you maybe can give us some insight on any challenges on technology needed to produce oligonucleotides at scale of production and getting this type of therapies to people, and maybe just in your experience, what has been some of the challenges that have been overcome for mass production?
The mass production of this type of chemistry at 20 nucleotides is much easier than making a messenger RNA by Moderna of 1,000 nucleotides. It's totally synthetic. It's totally on a column, and you run compounds through it. Scaling, I mean, we're using milligrams of material, so you can get a very significant scale. But the number of patients, just because of the low dose that you're delivering. So and the cost of oligonucleotides is much less than protein in terms of production of a large protein. So I think it's a technology that will be used broadly. The big issue is going to be delivered by injection. Injection either IV or sub-Q. Yes, we can do it orally, but it's not very efficient. Maybe it'll be more efficient.
Are there other questions?
I have a question about the looks like lumbar injection is pretty efficacious.
I say it again?
Lumbar injection,
Yeah.
it looks a lot of
Yeah.
to bring it
Yeah.
down, but it still has delivery issue. You worry about it? That's first question. And the second question is, did you understand first question?
Repeat it, please.
So lumbar
Yeah.
injection looks like a great efficacy, bring it down the gene expression, but you worry about the delivery issue is the main for siRNA field. So for all the CNS delivery, if it's worry about, so lumbar.
Well, lumbar,
Yeah.
I'm saying I think it's a significant advance that you can get oligonucleotides delivered in the CNS by lumbar delivery. And that I think it's an important step forward in silencing genes in the CNS. And it's still to be explored, but I think it's a really exciting idea. If we can develop, when we develop, similar specific delivery to other cell types in the body, then we have a number of other opportunities to treat and impact on disease. But these are two examples, liver and the CNS, that I think has had major advances.
Can I ask one more? Sure. So the second question is, you showing the siRNA, the chemical, the conjugate, right? Or we can use a lipid nanoparticle as well. So can you explain pro and con, either delivery system?
The nanoparticle is much larger. It has to be delivered, in this case, to get to the liver IV. And it has it's just a particle, and even though it's coated with PEG, there is some complement activation when you deliver it. So it's a great technology if it cures your disease and otherwise you're dead. But it's not as convenient because it has to be IV'd. Whereas the conjugate itself, you can deliver sub-Q. So anybody can self-injection. I mean, I've self-injected for other reasons. It's not an issue. You just stick a needle in your...
We have time for another. Yes.
I think there's a question over there.
It's great data with the delivery of the RNAs, but has industry been able to address the stability of the siRNA or the mRNA for commercial scale and use in the therapeutic areas?
The siRNA that's being used in the sub-Q delivery is chemically modified to be stable to nucleases in the bloodstream. And that is a big addition. And then the only thing that we discovered, in my opinion, as we developed this technology, is this long-term stability in cells. I mean, that RNA is in those cells for 6-9 months, silencing genes. And that's really striking.
I was thinking more
These don't have stability issues. They're as stable as chemical compounds are.
We have
And
one-
no cold temperatures.
Is the modification to the
Yep.
RNAs that
Yep.
make them stable?
Yeah, yeah. The LNPs are complicated because they're not modified, but these are different.
One last question.
How are the lipid nanoparticles or the siRNA conjugates engineered to avoid clearance so that they could be
Well,
delivered?
they are cleared from the system. RNA, once it's in cells, microRNA have a half-life of months. So it isn't that they're cleared once they're in cells rapidly. Once they're in the bloodstream, they come out through the kidney. It's mostly, or your intestine, and it's days. You see clearance within a day of most of the material that's not taken up by cells.
Thank you so much, Professor Sharp.